Hey everyone, I'm Callie, and today we're going to go over the rest of chapter 28 and 29, which is with fungi. And I was way too lazy to make a PowerPoint, so you're just going to have to look at my notes, and hopefully most of it's readable. Obviously, I'm going to discuss it, so let's get started. Just a reminder, for chapter 28, we break it down between green algae, abbreviated GA, and land plants. Land plants can be vascular or non-vascular, which means that they do or do not contain vascular tissue, which is used to transport water and nutrients. We break up vascular plants between seedless and seed producing, and then seed producing plants can be broken up into angiosperms and gymnosperms. But we'll get into those differentiations a little bit later. Why does the book justify grouping green algae and land plants together? This is definitely stuff that you need to know, the similarities. So the chloroplasts contain these sorts of pigments: chlorophyll A and B, and beta carotene, which causes things like carrots to be orange. Green algae and land plants have similar thylakoid arrangements. Thylakoids are part of a chloroplast. And then their cell walls, their sperm, and the peroxisomes are similar. And then both in green algae and land plants, chloroplasts synthesize starch for storage. And now we can talk about the difference between non-vascular plants and then vascular plants. So like I said, non-vascular plants lack vascular tissue. Those are specialized cell groups that conduct water and nutrients from one area to another. So because they don't have vascular tissue, they tend to be very short. They are reliant upon water in their environment. So things like mosses. And then for the reproduction in non-vascular plants, they use spores. Spores are single haploid cells which divide through mitosis and grows into a new organism for reproduction and dispersal. Now, when we talk about spores, that's just the tip of the iceberg. You're going to hear the word spores and, sporophytes and things like that many times, and it's really important in this chapter and in the chapter on fungi to not get those mixed up. But we're going to go through some strategies on how to differentiate the two, because life cycles can definitely be confusing. I know they can be kind of confusing for me. I'm sure some of you are confused by them. I really don't blame you. Okay, so for the vascular plants, they're either seedless or they have seeds. If they are seedless, they rely on spores. Some seedless vascular plants that we all know about include ferns. So a seed for a vascular plant who produces seeds contains an embryo and a store of nutritive tissue surrounded by a protective layer. The first seeds to appear in evolutionary history were the gymnosperms, which produce seeds and cones, some of them. Angiosperms are your flowering plants. That's a much larger group compared to the gymnosperms, very diverse group as well. And they have flowers and fruits to improve seed dispersal. And then we just kind of go into some evolutionary history, which I don't think you really need to know about, but it's kind of interesting. If we want to talk about some important factors of land plants, we talked about the cuticle. The cuticle is a waxy outer coating on a plant tissue to prevent water loss. I think that's all we need to know. This page, for some reason, is a little blurry, so we're just gonna have to get through this as best as we can. So next, we talked about The key factors that let plants transition to land. Why is it critical to recognize these features? Well, for plants, light and carbon dioxide are readily abundant on land. So it was beneficial for them to eventually evolve and have these mechanisms that allow them to live where these kinds of nutrients are more abundant. But to do that, they need to be able to prevent water loss. So like I said, they developed a cuticle, which is a waxy layer that prevents water loss from the stems and leaves. Then we talked about stomata, which have, they are essentially pores that allow for gas exchange in the photosynthetic tissues. This pore is surrounded by 2 guard cells, and based on the shape of the guard cells, the pore can open or close. And the pore closes at night to store the water, the carbon dioxide, and things like that, which are important for the Calvin cycle, which happens when light is not readily abundant, like at nighttime. And this is present in all land plants except for liverworts. Another important thing is resisting ultraviolet radiation. Plants produce flavonoids that absorb UV light to protect the DNA from being changed by ultraviolet radiation. We talked about lignin. Lignin is a defining feature of vascular tissue. It's really critical for a plant to stand upright because they have to transport water against the force of gravity. So, and they also have to be rigid enough to not completely fall over in a gust of wind. So plant cells became stronger over time because of lignin and the development of secondary cell walls. I don't think we talked about this in class, but tracheids, I don't know how to pronounce that exactly, but tracheids are another feature of what helps a plant stay upright. So they have a lignin-containing secondary cell wall, as well as a cellulose-based primary cell wall. They have pits in the sides and the ends of the wall where secondary cell wall is absent, and then it allows for efficient water flow. Another sort of structure are vessel elements. These are shorter and wider than the tracheids, and they have perforations. Again, I don't know how much we really even talked about that, but the significance of these sorts of structures is that they can form wood. A very big thing that we've been talking about are life cycles and reproduction. And this is where the chapter can get really vocabulary dependent, but I'm hoping that my many life cycle pictures will kind of help us go through things. So to just go through a lot of these vocabulary terms, we've got gametangia. It's a structure that protects the gametes, except in angiosperms. Antheridium is where sperm is produced. It's the sperm producing gametangia. Archegonium is the egg producing gametangia. Then when we talk about land plants, we have the concept of embryophytes, which means that embryos are nourished by the parental plant's tissues. Zygote retention and nourishment during initial stages of growth is critical to some plants so that they will have the most reproductive success. A big concept that we've been talking about is alternation of generations. This is where the multicellular haploid and diploid phases change. So gametophytes are the multicellular haploid phase. Sporophytes are the multicellular diploid phase. This is going to be a lot, I hope it's going to be a lot clearer once we talk about the actual life cycles. So down here is just a basic life cycle. So we start off with a diploid 2N sporophyte. Sporophytes produce haploid spores through meiosis. To get from diploid to haploid, you have to go through meiosis, which means that you're introducing genetic diversity into those spores. Then those spores can undergo mitosis to create a multicellular gametophyte, which is also haploid. That gametophyte can go through mitosis itself to produce gametes, again, also haploid. Then you can have a haploid gamete from one plant mix with another gamete, whether it's from its own structure, which would be self-fertilization, or from an outside structure, like if pollen were to be transported to that plant. Then you have fertilization, which produces a diploid zygote. Then over time, this zygote will undergo mitosis to form a multicellular diploid sporophyte. So to build the basics of your understanding of life cycles, it's really important that you understand the differences between diploid and haploid as we move along in a life cycle, and the ways in which you go from diploid to haploid. So remember, sporophytes are diploid. They produce haploid spores by meiosis. Gametophytes are haploid, and they produce haploid gametes by mitosis. It's also maybe helpful to understand whether or not some of these structures are multicellular or unicellular. Sporophytes and gametophytes are both multicellular, and then the things that they produce are unicellular. But those unicellular structures, such as the spores and the gametes, they can divide through mitosis down the line. In the case of gametes, they actually directly go from haploid to diploid, forming a zygote through fertilization, but then that zygote can undergo mitosis to form a multicellular structure that we know as the sporophyte. Then this is kind of cut off. It says heterospory. This means that a plant produces two distinct types of spores. So they produce eggs or sperm, and when we're talking about heterospores, we're talking about seed plants. So that's the vascular plants that have seeds. Homospory means that a plant produces a single type of spore, egg and sperm. This is the non-vascular plants and the seedless vascular plants. Now, adding on to the concept of being heterosporous, so we're dealing with the seed plants here, we have microsporangia, megasporangia, and the different features that those produce. Microsporangia produce microspores, which develop into male gametophytes, and then those male gametophytes produce sperm by mitosis. So remember here, microsporangia produce microspores. Those microspores develop into male gametophytes, which produce sperm mitotically, which is also known as a pollen grain. Megasporangia produce megaspores that develop into female gametophytes, and those produce eggs. So megasporangia produce megaspores that develop into the female gametophyte. Pollen grains form in heterospore seed plants, like we've been talking about, where the microspore, again, the male producing structure. So the microspore germinates to form a tiny male gametophyte surrounded by sporopollenin. Talking about the megasporangia and the female side of the plant, the plant and its reproduction, seeds develop from an ovule, which is a megasporangium with one or more eggs, and have an embryo and food supply with a tough coating. Again, I have a ton of life cycles later on that hopefully should clear things up a little bit. The more life cycles that you look at and are able to draw yourself really helps. And so when we're trying to differentiate between microsporangia and megasporangia and what produces female and male gametes in a flowering seed plant, we talked about some different structures, such as the stamen, which has the anther, which is where microsporangia develop. So this is where we can eventually find our male gametes. Meiosis occurs in the microsporangia. These microspores divide by mitosis to form pollen grains, which are male gametophytes. The carpel, that focuses on an ovary where the ovules are found. Meiosis occurs, forming megaspores within megasporangia. Megaspores divide mitotically, forming female gametophytes. And an interesting thing about how the plant supports a developing embryo is actually that double fertilization occurs, which produces, it's not haploid, it's not diploid, it produces a 3N nutritive tissue called the endosperm to support the diploid zygote as it becomes an embryo and grows. Looking and seeing what is relevant. Fruits are derived from ovaries and enclose one or more seeds. Fruits are important for seed dispersal. They are very desirable by animals, so the animal eats it and transports the seeds through its gastrointestinal tract elsewhere to where the seeds can hopefully turn into a plant. So another key factor about angiosperms is how diverse they are. I think we talked about adaptive radiation maybe once. It means a single lineage produces a large number of descendant species, which are adapted to a wide variety of habitats. Basically just explaining and reiterating how diverse angiosperms are. And then we talked about monocots and eudicots. The book goes into a long explanation as to why this name for the eudicots went from dicots to eudicots. So what you need to know for this is that this is one way that we can differentiate the angiosperms. So, and again, I have no clue how to. Some of these words are not easy to pronounce. Cotyledons. I'm not the kind of person who can pronounce things very well, but that's also referred to as a seed leaf, and it's a place where nutrients can be stored to supply to a developing embryonic plant. So it refers to the first leaf that a plant will have, which is really crucial in supporting the plant's growth. Monocots have one seed leaf. Eudicots have two seed leaves. And by breaking these two groups down, we can see that they have some interesting factors. So in monocots, vascular tissue is scattered throughout the stem. So if you take a cross-section of a plant stem, if it's a monocot, you'll see the vascular tissue spread out kind of like chocolate chips on a chocolate chip cookie. Maybe I'm just saying that because I had a cookie earlier. I don't know, that's what it looks like. Their leaves will be parallel. So basically just some parallel lines. That's definitely something you can see easier if you are out and about and you look at leaves. And then their petals also come in multiples of 3. Now the eudicots have 2 seed leaves. Their vascular tissue in the stem has a circular arrangement. They have branching veins in the leaves, which now that it's fall time and leaves have fallen, that's pretty easy to see. If you go and pick up some leaves and just take a look at them, you can see that their veins are branching out. And then their petals are in multiples of 4 or 5. And then with the lichens, we'll get to that in fungi. Rhizoids, this is how some plants can be anchored to soil, rocks, or tree bark. Okay, so here's the first life cycle that I have. This is for this is for the seedless vascular plant. And this has, is this a seedless vascular? Yes. I'm trying to see what this order is. Actually, let me go up here. Let's just talk more about the non-vascular and the seedless vascular plants one more time before we get to the craziness that is life cycles. So non-vascular plants such as mosses, corn, and liverworts are low sprawling, tied to wet environments because they don't have vascular tissue. They don't have true roots. The rhizoids are the anchor to whatever they are attached to. No true vascular tissue. They're gametophyte dominant. The sporophyte's attached to the gametophyte, which is actually right here. The sperm has the flagella, and their spores are dispersed by wind or rain. Seedless vascular plants include club mosses, ferns, and horsetails. They're sporophyte dominant. They grow from, the sporophyte grows from the gametophyte. They have vascular tissue with lignin. The sperm has a flagella, and they are water dependent on reproduction. Okay, so here is an example. I think this was actually in the book where I got this life cycle from, but essentially on the same plant, you have both the female gametophyte and the sporophyte. So the mature sporophyte is diploid. By meiosis, it will produce genetically diverse spores, which are haploid. These spores will be dispersed. Then the spore can divide mitotically, and it will either develop into the male part of the plant or the female part. You can see this. It has essentially 2 pathways it can go down. Now, if it is a male plant, you'll have the sperm being developed in the antheridia, which is on the top of this particular plant. If it's a female gametophyte, then you'll have eggs developing in the archegonia. The sperm swims to the eggs, you have fertilization. Now you've got a diploid zygote housed in the archegonium. Through mitosis, you have this structure develop into a diploid sporophyte, and then eventually it just, it continues going. Here's another example with ferns. So you start off with a mature sporophyte, which is diploid. It undergoes meiosis to produce genetically diverse spores, which are housed in sporangia. These are dispersed by the wind. These spores undergo mitosis, to create multicellular haploid gametophytes, which are over here, these heart-looking things. And then these continue going through mitosis to mature into mature gametophytes. For this particular example, it says that sperm and eggs develop at different times, so self-fertilization is produced. And again, it's important to remember sperm develop in the antheridia, eggs develop in the archegonia. Once the sperm swims through the egg, you have fertilization. Now the zygote's housed in the archegonium. And here's just a picture of where the gametophyte and the sporophyte are located. Here's a good review about the difference between homospory and heterospory. Homospory produce the gametophyte produces both egg and sperm. Heterospores, the gametophyte can produce egg or sperm, which led to seed and pollen evolution. And then, of course, seed plants, which have micro and megasporangia. Whereas if we go back to the homospores, if the gametophyte produces egg and sperm, you have a self-fertile plant, and these lead to the this classification being for the non-vascular plants and the seedless vascular plants. Now, if we want to talk about our vascular plants with seeds, we've got the gymnosperms. So you start off with a mature sporophyte, which is diploid. It produces two structures. It produces cones that have microsporangia. Remember, if we go down this line, we're talking about the male structure on the plant, which produces sperm, and then it also produces an ovulate cone. If we go down this route, we're talking about the female side, which produces eggs. If we go back to the male side with a microsporangia, this will undergo meiosis to produce haploid microspores. These will undergo mitosis, forming the male gametophyte. And then if we go back to the ovulate cone side, it forms ovules containing megasporangia. Then we've got a little diagram of a megasporangium. And then this can divide through meiosis. And then eventually we will have pollination, where the sperm will enter this the structure, the megasporangium. Mitotically, it can divide, and now you have a diploid embryo. Eventually, these will form these little, I think these are what people say are helicopters. You can see them often. It's just designed for them to be carried through the air pretty efficiently. So these seeds are dispersed in the wind, or via a vector, like an animal, if an animal eats it. And then if it lands, then it can eventually grow and develop back into a mature diploid sporophyte. Now, if we're talking about flowering plants, so there's a couple different structures here. So the part in the middle is the carpel. When we're talking about carpel, we're talking about where you can get eggs from. And then we've got stamen. The top of a stamen is an anther. When we're talking about that, we're talking about the male side of the plant where you get sperm from. So anthers produce microspores. They can divide through mitosis to form the male gametophyte, the pollen grain. In the carpel, we have an ovary, which contains a megasporangium housed in an ovule. Through mitosis, we will have megaspores, which can grow into female gametophytes inside the ovary. And then we will have the sperm pollinate the plant, entering this the carpel, this structure here, fertilizing this egg. And this is where we get double fertilization. So a sperm will fuse with the egg to produce the diploid zygote, but then we can also have a sperm fusing with 2 nuclei to produce a 3N endosperm. That's the nutritive tissue that this zygote can use. And then over time, this structure will divide mitotically to form a fruit. Then inside that fruit, of course, you have seeds, which will hopefully be dispersed in a wide variety of ways. And then hopefully it will grow into a sporophyte. And again, all land plants undergo alternation of generations. So we go from a multicellular diploid sporophyte, we go through meiosis, producing haploid spores, which undergo mitosis to produce a haploid multicellular gametophyte that can go through mitosis as well, forming, again, unicellular haploid gametes. Gametes can fuse and undergo fertilization, producing a diploid zygote, which then can mature and divide into a multicellular diploid sporophyte. So essentially, this understanding how we get from sporophyte to spore to gametophyte to gametes to zygote and back around is really critical in making your way through these often really confusing life cycles. Okay, so now just some quick things about fungi. When we talk about mycorrhizal, that's the fungi that live in close association with plant roots. The relationship that fungi have with plants is extremely important. And from our class, we've talked about how oftentimes we see plants benefiting from these interactions with the fungi, with the mycorrhizae. So fungi are important because they break down a lot of dead tissue, allowing for the nutrients inside that dead tissue to be used and be available by other organisms, primarily plants, of course, since plants are primary producers, which then can provide food, nutrients, etc., for other organisms. Saprophytes are fungi that digest dead plant materials. They're recyclers, very critical to the carbon cycle. They are connected due to the fixation of carbon cycle by land plants, and then the release of carbon dioxide due to cellular respiration. So again, fungi are great recyclers, breaking down waste products and returning those trapped nutrients back into the environment for other organisms to use. We talked about 2 growth forms. We've got single-celled yeasts, and then we have multicellular filamentous mycelia. The filaments that make up a mycelium are called hyphae. Each filament can be divided into cells by cross walls called septa, and these walls don't have to be completely blocking off the rest of the hyphae. There can be pores to allow for nutrient transport, but some fungi actually don't have these walls, the septa. Those kinds of fungi are called cenocytic. They actually have many nuclei inside their hyphae. So fungi reproduce using spores. Oops, let me go back. Okay, so there's a lot of terminology when it comes to To how fungi reproduce, of course, they're very diverse, just like plants are. So here's a couple ways. You got swimming gametes and spores. Zygosporangia, that's the haploid hyphae form from two individuals joined to form a spore-producing zygosporangium. Again, I have life cycles of all of these, so don't worry just yet. Basidia they have club-shaped cells at the ends of the hyphae, and each basidium produces 4 spores by meiosis. The asci, I'm terrible at pronouncing these things. This is the worst. Anyways, they have sac-like cells at the tips of the hyphae, in each ascus. Meiosis and 1 round of mitosis produce 8 spores. Then we have the conidia, which are asexual spores that can be dispersed, such as mold, which we've probably all seen. Interestingly enough, fungi are closely related to animals more so than land plants. We talked about a couple ways that we see this. Chitin production, the shape of flagella is similar, and then fungi synthesize glycogen for storage. Remember that plants use starch for storage. And again, fungi are super important because they often have symbiotic relationships. It can be mutualistic, where both organisms benefit, parasitic, where one organism benefits at the cost of the other one being harmed, and then a commensal relationship is where one organism benefits and the other organism is not affected. More cool ways about how fungi interact with plants, we've got our ectomycorrhizal fungi. These fungi will form some very significant sheaths or like coatings around a plant's roots, and then they can get in between the root cells. Our arbuscular or endomycorrhizal fungi can actually penetrate a plant's cell wall, helping to transport nutrients to that plant. And then if we're talking about organisms that live between and within plant cells, it's called an endophyte. And these endophytes are usually above ground. Whereas if we're talking about ectomycorrhizal and endomycorrhizal, we're talking about often the roots. So here's just some general guidelines on fungi life cycles. Just some terminology to know. Plasmogamy is when hyphae from 2 different mating types grow near each other and fuse to form a hybrid cell. Heterokaryotic is where genetically distinct haploid nuclei persist in the same cell or the mycelium. Dikaryotic is where most hyphae have their cells divided by septa, those walls again, and each cell contains 2 nuclei, 1 from each mating type. So some really interesting stuff that we haven't seen yet, and that we'll get to in just a minute. The book provides a generalized life cycle for both ways that asexual and sexual reproduction, 2 ways that the mycelium can reproduce. For asexual reproduction, we've got it dividing through mitosis with a spore-producing structure that's formed, which can also divide again mitotically to produce spores, and we can continue on in this asexual side. If we get to the sexual reproduction side, we've got our plasmogamy, which is when 2 hyphae fuse to form a hybrid, producing this heterokaryotic mycelium. And you see it's not 2n, it's actually n plus n, which is a distinction to show that this is a hybrid cell. Again, heterokaryotes, we're talking about genetically distinct haploid nuclei in the same cell, in the same mycelium. Then with karyogamy, we produce actually, let me go over karyogamy. That's just the fusion of the nuclei to form diploid zygotes. So you eventually take these and they're actually going to fuse to form a diploid zygote, which can go through meiosis to produce haploid spore-producing structures. Then we have spores, and again, it returns back to a mature mycelium, which is haploid. And then the book describes a few key lineages of fungi and their life cycles, which can be vastly different. So here's a chytrid. I know I'm going to get a lot of these wrong, and I'm sorry, but it's just, it's the way that biology goes, I guess. So we're talking about this specific structure on the mycelium, which is where gametes are formed. You see, we've got the male and the female gametes. These fuse. You have plasmogamy and karyogamy. Let me go back again because these are really important. So again, we've got the hybrid fusing to form a diploid zygote, dividing mitotically to form this really interesting structure where this is a sporophyte mycelium, which is diploid. The spores are formed in the sporangia, and then it continues on with the haploid spores, which can divide mitotically again to return it to a gametophyte mycelium. I'm probably going to really butcher this. Zygomycetes life cycle. So this is probably one you may have been familiar with. You might have seen it. So we have asexual sporangium, which produce conidia. Here we've got those filaments, those hyphae of different mating types. These are different organisms fusing. And then we go through plasmogamy. So you can see that the hyphae have fused. In between those hyphae where they meet, we've got this hybrid. The spores form in this zygosporangium. You can see those little dots that I've drawn. And eventually these will fuse to produce a diploid zygote, which can then divide through meiosis to go back to a haploid stage, and then that can go through mitosis to return it back to this mature fungi. Okay, I think this one got cut off a little bit. This one is with Basidia. So this is, a pretty typical mushroom. So here's a structure called a basidium. On the end is where there are spores. You have karyogamy, where these are fusing to produce a diploid, which goes through meiosis to produce haploid spores. These spores can be dispersed by the wind. Then these spores can divide mitotically to produce this larger structure. These spores germinate to form hyphae. And then we've got another period of plasmogamy where we return to it being a hybrid, and then this can grow. With Ascomycota, so the structure we have here is a mature ascus and a young ascus. We see this as N+N. So we're going to have karyogamy, the fusion, producing a diploid. This diploid structure goes through meiosis to produce a bunch of haploids, 4 to be precise here, which can divide through through mitosis, and then these haploid spores are dispersed through the wind. Then these spores can germinate again to form hyphae. Then we have the situation where we have many nuclei. Then we can go through plasmogamy, and we can see that this dikaryote, this N+N mycelium, can grow and then return to the mature fungi through mitosis. And then here's just some brief notes that I took from class. Fungi are not photosynthetic. There's a ton of different things that we commonly call fungi. You know, you have your molds, mushrooms, your mildew. This should be mildew. Rusts, blights, wilts, and yeasts. Again, they're super important in ecosystems because of their relationship with other organisms. They're eukaryotes, heterotrophs, saprophytic. They're cyclers that excrete enzymes to break down material. They're decomposers, and they can be single-celled yeasts or multicellular myceliums. Here's again some connections with animals. We've got the chitin and the glycogen versus the starch in the plants. Multicellular fungi have hyphae, which are very thin filaments, which are dependent on water. They can have those walls, the septa, or they can be seen acidic, where they have lots of nuclei in the hyphae. These hyphae can be extremely large. And then one last term that we discussed was lichens, which is a mutualistic, with both organisms benefiting, relationship between a fungus and a photosynthetic organism, such as a cyanobacterium or green algae. And then one last thing to know about life cycles for fungi is that they can be asexual or sexual reproductors, and they do not undergo alternation of generations. So I hope that was everything. That was a lot. I'm sure I made some sort of mistake, but I hope that this is helpful, and good luck on the exam.